Afternoon, everyone. Welcome to lecture 3 on cell cycle and cell division. So, in today's lecture, we will be continuing with what we left with the last. So, in the last lecture, we discussed about mitosis. So, just for a quick revision, so mitosis is also known as equatorial division. In this, a single cell, which is 2n in number, will give rise to cells which are similar in genetic composition and which will have the same number of chromosomes. So basically, there is the genetic similarity between the parental cells and the daughter cells. So these two are genetically similar. So they produce genetically similar cell. So the complete M phase that involves the mitosis is divided into 4 phases that we see in the last lecture. So it is prophase, metaphase, anaphase, and telophase, which is further followed by cytokinesis. All these steps results in karyokinesis, that is the division of nucleus into two. And cytokinesis finally results in the separation of those two nucleus into the 2 different cells. So this was what we understood about the mitosis in the last lecture. But then there is another kind of division, that is the meiosis, that we will start in today's lecture. So we know that during the time of reproduction, so there is a formation of gametes. So the gamete formation results in the production of sperm and ovum, for example, and the fusion of sperm and ova results in the formation of a zygote. So how does a diploid organism, which is already 2n in number, maintained its number in the zygote even after the fertilization? So here the process of fertilization happens. So even after the fusion of sperm and ova, how does in zygote we are able to maintain the same chromosome number? So this happens by the process of meiosis. So the process of meiosis is also known as reductional division. It leads to the reduction in the number of chromosomes. So starting with the 2n organism, in the sperm and ova, we will have only the haploid state, that is only the n number of chromosomes. And upon fertilization, in which when the zygote is formed, again it will be a 2n number. That is, again a diploid organism is formed. So the production of offsprings by sexual reproduction includes the union of 2 cells. That is, sperm and ova fuses, right? Each with a haploid set of chromosome. So the haploid set of chromosome fuses to again give rise to the diploid cells. So the doubling of the chromosome number at fertilization is compensated by an equivalent reduction in the chromosome number at a stage prior to the formation of the gametes. So as I mentioned, like for example, if I have to tell this, so if, for example, we start with an organism which is 2n in number. Now, this is male and it has to be fertilized with some female, which is also 2n. And suppose there is no reductional division and there is a fusion happens between the male and the female gametes. Considering the fusion happens, so the zygote that will be formed without any division at this stage, without meiosis. This is without meiosis. So what will happen? The zygote should have 4n number. But this is not the case. As I mentioned, when there is a fusion between the male and the female gametes, that is the sperm and the ova. So during the formation of sperm and ova, the process of meiosis takes place. Process of meiosis takes place during the formation of gamete. That is, during the gametogenesis, the process of meiosis happens. resulting in the sperm and ova, which is half the number of chromosomes than the organism. Now, upon fertilization of this sperm and ova, so this is the fusion or the fertilization that happens that results in the formation of zygote. Now, this zygote will be what? It will be again 2n in number. So, basically, this diploid state at the zygote level is maintained or is possible due to the reductional division or the meiosis that happens during the formation of gametes. So, this is the importance of meiosis, that why meiosis is important, because in certain stages where we need to have the organism of the same number of the chromosomes, like during the sexual reproduction, where there is a fusion of the gametes, then the gametes need to be first developed into the haploid state so that their fusion results in the diploid zygote. So this conversion of diploid to haploid is taking place with the help of the meiosis, which is also known as reductional division. So the doubling of the chromosome number at the fertilization. So basically, they are talking about this situation where the doubling of the chromosome number may happen during the fertilization, but this does not take place because of the process which is known as the meiosis. So this is compensated by an equivalent reduction in the chromosome number at a stage prior to the formation of the gametes. So this is happened by the process which is known as meiosis. This meiosis term is coined in the year 1905, and it has been originated from the Greek word that means the reduction. So therefore, it is also known as reductional, since it leads to the reduction in the number of chromosomes. Therefore, it is also known as reductional division. Meiosis ensures production of a haploid phase in the life cycle. So wherever there is a conversion of diploid to haploid, this happens by the process which is known as the meiosis. So meiosis ensures the production of a haploid phase, and fertilization ensures a diploid phase. So basically, fertilization results in the diploid phase and the meiosis results in the haploid phase. So whenever there is a haploid phase that is to be formed or that is required, that will happen by the process of meiosis, and upon fertilization, these will again be converted into a diploid phase. So as I already told in this, that this diploid to haploid conversion happens by the process of meiosis, and upon fertilization, the formation of zygote again results in the formation of a diploid phase. So without meiosis, what will happen? The chromosome number would double with each generation. So suppose, as I mentioned in this example, so in this, the first generation, after this, the first generation would have the 4n number of chromosomes. Now, when these 4n number of chromosomes will fuse without any process of meiosis, it will result in the 8n number of chromosomes. Therefore, the chromosome number will keep on increasing with every subsequent generation, and therefore it will be difficult for the organism to maintain the chromosomal state, which is the like for example the diploid chromosomal state. So therefore the sexual reproduction would not be possible further without the process of meiosis. So this is the importance of meiosis, that why we need the meiosis, why do we need to reduce the number of chromosomes. So meiosis, unlike mitosis, where we do not have any mitosis 1 or mitosis 2, in meiosis, we have two types of meiosis 1 and meiosis, which is followed by meiosis 2. So meiosis 1 results in the segregation. So in meiosis 1, the homologous pairs are separated, reducing the chromosome number by half. So the chromosomal reduction happens in the meiosis 1, while in meiosis 2. So meiosis 2 is similar to what we have understood in the process of mitosis, where the sister chromatid separation happens and resulting in the 4 haploid gametes. While in meiosis 1, there is the segregation of the chromosome. So we know that even in our cells, we have 2 chromosomes, right? We have 2 chromosomes. So in meiosis 1, there is a separation of these chromosomes, And then after this, we have the production of 4 haploid gametes. So this happened by the meiosis 2, where there is a segregation of sister chromatids, and the segregation of sister chromatids result in the production of the 4 haploid gametes. So as with mitosis, the prelude to meiosis includes DNA replication. So firstly, DNA replication has to happen, and the premeiotic, that is the phase which is before the meiosis. So the premeiotic S phase generally takes several times longer than a premitotic S phase. Right? So prophase of the first meiotic division, that is the prophase 1, is typically lengthened in extraordinary fashion when compared to mitotic prophase. It comparatively takes larger time as compared to mitosis. So meiotic prophase is further divided into the 5 stages, which is leptotene, zygotene, pectine, diplotene, and diakinesis. So this you need to remember. These are the 5 stages of the prophase 1. So in meiosis takes place in 2 stages, meiosis 1, meiosis 2. In meiosis 1, segregation happens. Segregation of what? Segregation of homologous chromosomes, right? While in meiosis 2, there is a segregation of sister chromatids, right? So in this meiosis 1, again, it has the similar type of stages: prophase, metaphase, anaphase, and telophase. So the prophase 1 of the meiosis is divided into 5 different stages: leptotene, zygotene, pectine, diplotene, and diakinesis. So we will understand about these stages one by one. So what happens in leptotene? The chromatin threads condense to form the chromosome. So in our cell, usually when there is a non-active or form, so it is present in a thread-like form, which is the chromatin. This chromatin is nothing but the uncondensed form of the chromosome. So the chromatin threads condenses to form the chromosomes, and chromosomes are longest and thinnest at this stage. So this might be asked that the chromosomes are longest and thinnest at which stage? So they are longest and thinnest at leptotene stage. Now chromosomes become gradually visible under the light microscope. All the chromosomes in nucleus remain directed towards the centrioles. So group of chromosomes in nucleus appear like a bouquet. Therefore, it is also known as bouquet stage. So it's like all the chromosomes are towards the centrioles, and therefore they form a bouquet-like structure, and therefore it is known as a bouquet stage. The next stage is the zygotene or synaptotene stage. So it is characterized, as the name suggests, zygote. We know it is produced by the fusion, right? Or synapse means joint, any joint, right? So it is characterized by pairing. The name itself suggests what happens in this stage is pairing of homologous chromosome. That is the formation of synapses. So the parental chromosome and the maternal chromosome. So we have 2 chromosomes. One. So chromosome 1, these are both the chromosome 1 which we have obtained on the side, right? So during this, there is a pairing of the homologous chromosome. All the homologous chromosomes, like 1 forms a pair with 1, 2 forms a pair with 2, 3 forms a pair with 3, etc. So all the homologous chromosomes forms a synapse, and it is characterized by the pairing of homologous chromosome. A structure develops in between the homologous chromosome, which is termed as the synaptonemal complex. So the structure which is developed by the fusion of these 2 homologous chromosomes is known as the synaptonemal complex. And the pair of homologous chromosomes are called as bivalents or tetrads. So you can see here, these are called as bivalents now, or they may be called as tetrads because of these 4 arms, right? So they are more clearly visible at the next stage, pectine. The first two stages of prophase 1 are relatively short-lived compared to the pectine. So pectine is a longer stage, and during this stage, the four chromatids of each bivalent chromosome become distinct and clearly appeared as tetrad. So you can see here that what happens in this is, so this is chromosome 1, this is also chromosome 1, but with this, they have one sister chromatid attached to it. They also have one sister chromatid attached to it, forming a tetrad. 4 arms. So they have 4 arms forming a tetrad, and this tetrad is clearly visible in the pectine stage. Recombination nodules between non-sister chromatids. So in mitosis, we see that they were called as sister chromatids as they are the chromatids of the same chromosome. But in this, suppose this is the one, this is also one. This is from the paternal side, this is from the maternal side. Now the sister chromatid of this paternal with the maternal is called as the non-sister chromatid. So this is very important. In this, the sister chromatids, in this, recombination nodule is formed between the non-sister chromatids. This recombination is important because this recombination or the exchange of genetic material at this stage leads to the variation, which is absent in mitosis. Right? So the recombination nodules is very important in this concept because meiosis, as I firstly mentioned, it is a reductional division. It helps in maintaining the chromosomal number in the zygote stage, but at the same time, it is also helping in creating the variation by the process which is known as the recombination that happens during the prophase 1 stage of the meiosis 1. So recombination nodule is formed between the non-sister chromatids of the homologous pair developed, and these non-sister chromatids exchange their parts, that is the crossing over. So these non-sister chromatids exchange their genetic material, which is known as the crossing over, and the process is known as the recombination. Crossing over leads to recombination of genetic material. So the genetic material of this one DNA was, so if I have to tell you, so suppose this is the one non-sister chromatid and this is the other non-sister chromatid. So the crossing over happens in between these, and so if I have to show them differently, let's say this is one and the crossing over happens in between them. So what will happen is there is a change in the genetic composition. There is the exchange in the genetic composition. So what will happen is now these will have certain genetic material of certain genetic material of the other non-sister chromatid. So this is how recombination or crossing over happens. So crossing over leads to a recombination of genetic material on the two chromosomes, and it is an enzyme mediated process, and the enzyme which is involved in this is recombinase. Since the process is recombination, therefore the enzyme is termed as recombinase enzyme. Recombination between homologous chromosome is completed by the end of pectine stage, leaving the chromosomes linked at the site of crossing over. So the chromosomes will remain linked at the site where the crossing over has taken place. So the beginning of diplotin is characterized by the dissolution of synapse complex. Homologous chromosomes start sequence. Such structure appears during the diplotin stage. So before that, both the homologous chromosomes are linked together, but the beginning of the diplotin stage is characterized by the dissolution of the synaptonemal complex. So the synaptonemal complex dissolves and it results in the repulsion of the both the chromosomes from each other, so that the X-shaped structure appears. So both the chromosomes repel each other and an X-shaped structure is formed. This is known as the meta. So these terms are important. Meta is formed during the protein stage, while recombination happens in the peptide stage. So these stages are important. So like chitin, the X-shaped structure called chiasmatic is formed, and in certain cases, for example, in the oocyte of some vertebrates, it may last up to the months or the years. The next stage is the diakinesis. So it is a final stage of meiotic prophase 1. It is marked by the terminalization of chiasmata. So chiasmata opens in a zip-like manner and chromosomes are fully condensed and meiotic spindle is assembled to prepare the homologous chromosome for separation. Centrioles move towards the opposite poles. By the end of diakinesis, nucleolus disappears and the nuclear envelope also breaks down. So now the cell is preparing itself to separate both the non-homologous chromosomes. That is, for the segregation of the non-homologous chromosomes, the cell is preparing itself, and by the end of the diakinesis, which is the last stage of the final stage of the meiotic prophase 1, the cell starts preparing, the nucleolus disappear completely and the nuclear envelope also breaks down, which will further facilitate the separation of the homologous chromosomes from each other. Now, diakinesis represents transition to the metaphase. So after this, what will happen in the next stage is the metaphase. So we can see in this figure, so this is the centromere. We know the chromosomes remain attached to the centromere in the leptotin stage. The chromosomes condense. Then in zygotine stage, there is a formation of synaptonymal complex. The homologous chromosomes, like these are the homologous chromosomes, they may be two. So the homologous chromosomes are come in contact with each other, forming a synaptonymal complex. This synaptonymal complex consists of the tetrads. That is, it has two sister chromatids. So these are the sister chromatids. These are again the sister chromatids, while this and this are the non-sister chromatids. So it consists of 4 arms, therefore it is a tetrad structure, and these two are the non-sister chromatids. This is chromosome 1, this is also chromosome 1, for example. These two are the sister chromatids. So in the next stage, which is the diplotin stage, the chiasmata, which is the cross-like structure or which is the egg-shaped structure, forms. So this chiasmata, which is the egg-shaped structure that is formed, results in the diplotin stage. So during this, what happens is the recombination. So in the chiasmata, recombination happens. So recombination happens by the process of chiasmata formation. So during the formation of chiasmata, we can see that there is a interaction between the non-sister chromatids. So you can see here that there is an interaction between the non-sister chromatids. The chromatid from one homologous pair interacts with the chromatid of the same homologous pair, and therefore the recombination happens. So the genetic material gets exchanged and the recombination happens. After this, in the diakinesis, there is a dissolution of the nucleolus and the nuclear envelope also starts disappearing, marking the next stage, which is the metaphase 1. So we know that in the metaphase, the two homologous chromosomes of each bivalent are connected to the spindle fibers from the opposite poles. So these are the two homologous chromosomes. So this is, for example, this is homologous chromosome 1 with 2. This is also homologous chromosome. It's homologous, which is the 1. This is 2. This is also 2. So these get attached to the spindle, opposite spindle pole. So let's say this is pole 1. This is pole 2. So these two get attached to the two different spindle poles so that in the next phase, these two can migrate towards the opposite pole. So in contrast, you can see that the sister chromatid are still connected to the tubules from the same spindle pole. So the sister chromatids, however, a chromatid pair. So sister chromatid, however, are attached to the same spindle pole. So therefore, there is no separation of sister chromatids. So sister chromatids does not get separate, does not separate during this phase. Because they are attached to the same spindle pole, only the homologous chromosomes will get separate. So sister chromatids, they are attached with side-by-side arrangement of their kinetochores. The orientation of the maternal and paternal chromosomes of the metaphase 1 plate is random. The maternal member of a particular bivalent has an equal likelihood of facing either pole. So it is not the paternal chromosome will face this side and the maternal will face this side. It can be anyways, and it is completely random. So therefore, this will also result in more variation, because an organism can have certain pair from the cells can have certain from the chromosomes from the parental side and certain from the paternal side. The next is the anaphase 1. So the separation of homologous chromosomes at anaphase 1 requires a dissolution of the chiasmata. So the chiasmata are maintained by the protein cohesin. Therefore, this cohesin is present between the sister chromatids in regions that flank the sites of recombination. So the chiasmata disappear at the metaphase 1 anaphase 1 transition, and the arms of the chromatids lose the cohesion. The loss of cohesion between the arms is accomplished by the proteolytic cleavage of the cohesin molecules. So basically, the how this chiasmata dissolution happens by the dissolution of the protein, which is the cohesin protein, and the enzyme, and it is a proteolytic cleavage where the cleavage of the protein happens by the enzymes, that is proteases. In contrast, cohesion between the joint centromeres of sister chromatids remains strong, because this cohesin is situated in between the sites, that is protected from the proteolytic attack. So it is very important that the cohesin protein, which is present between the homologous chromosome, that is the non-sister chromatids, gets dissolved, while if I talk about the cohesin protein between sister chromatids of the same chromosome remain intact. Right? So this is very important, that the cohesin protein between the sister chromatids remain intact, while the cohesin protein between the non-sister chromatids gets dissolved. So as a result, sister chromatids will remain attached to each other, while the homologous chromosomes will get separated. So consequently, when homologous chromosomes, let me visit. So consequently, when homologous chromosomes separate during anaphase 1, each pole receives a random assortment of maternal and paternal chromosomes. As I mentioned, that it is not like the paternal chromosomes will attach at the same pole and the maternal will attach on the other pole. Psychological event that corresponds to the Mendel's law of independent assortment. This is very important. The Mendel's law of independent assortment can be easily seen in the anaphase 1 of the cell cycle. As a result of independent assortment, organisms are capable of generating a nearly unlimited variety of gametes that may have some combinations of paternal chromosomes, some combination of maternal chromosomes, etc. So this is the metaphase 1 to anaphase 1 transition. You can see here that these are the to the opposite spindle poles. The cohesin protein between the sister chromatids, you can see here the cohesin protein between the sister chromatids remain intact, but the cohesin protein between these two homologous chromosomes gets dissolved, resulting in the separation of the homologous chromosome. So this results in the separation of the homologous chromosome, and you can see here this is the result of recombination, where there is an exchange of the genetic material between the non-sister chromatids. Right? Then the anaphase 1 will be followed by the telophase 1. So telophase 1 produces less dramatic changes than the telophase of mitosis uh, as now, because it has to undergone dispersion, they do not reach the extremely extended state of the interphase nucleus. The nuclear envelope may or may not reform during the telophase, called the interkinesis, and is generally short-lived. So between two meiotic divisions also, the stage is called as the interkinesis, and this interkinesis is short-lived as compared to what we see in the mitosis. So these are the 4 different stages of meiosis 1. So in the prophase, we see that there is a formation of Chi's matter, there is recombination, homologous chromosomes come in contact with each other, and in the metaphase 1, these homologous chromosomes align randomly at the opposite poles, and in the anaphase 1, the homologous chromosomes move to the opposite pole of the cell. So some part of, like, one pair of chromosome will move here, the other will move on the opposite poles. And in the telophase 1, which may or may not, like, which is followed by cytokinesis, the chromosomes gather at the poles of the cells and the cytoplasm divides, resulting in the formation of 2 cells that have the half and half number of the chromosomes. So the homologous chromosomes get separated. There will be one pair of chromosome in the first half and the another in the second. Now starts the stages of the meiosis 2. So the interkinesis, which is the stage between the 2 meiotic phase, is followed by the prophase 2. So this prophase 2 is much simpler as compared to the prophase 1. In prophase 1, we see that it has 5 stages where it undergoes the chiasmata formation, recombination happens, so there is some exchange of genetic material. But in this, it is quite simple. It is like the prophase of the mitosis. If the nuclear envelope, in case, had reformed in the telophase 1, it has to be broken down again. So what is actually happening in meiosis 2 is the separation of sister chromatids, right? So that is happening in the meiosis 2. So in meiosis 2, there is a separation of sister chromatids. So prophase 2 is followed by the metaphase 2. The chromosomes become recompacted and line up at the metaphase plate, just like what happens in the metaphase at the mitosis. And unlike metaphase 1, the kinetochores of sister chromatids of metaphase 2 face opposite poles. So in metaphase 1, we know the kinetochores of sister chromatids same pole. But in metaphase 2, the kinetochores of sister chromatids face the opposite poles and it become attached to the opposing sides of the chromosomal spindle fiber, therefore preparing themselves to separate the sister chromatids. In the anaphase 1, anaphase 2, it begins with the synchronous splitting of the centromeres, which had held the sister chromatids together, therefore allowing them to move towards the opposite poles. So basically in this, the separation of sister chromatids happen that were earlier present together. Separation of sister chromatids. Right? So in this anaphase 2, which is similar to the mitosis anaphase, in this the separation of sister chromatids happen and both the sister chromatids move towards the opposite poles of the chromosome. So meiosis 2 ends with the telophase 2, in which the chromosomes are once again. So as I mentioned, now there will be no term like chromatids. Now they will be called as the chromosomes. These chromosomes are once again enclosed by a nuclear envelope, and the product of meiosis are now haploid cells with 1C amount of nuclear DNA. So they have half the nuclear DNA. This is metaphase 2 and anaphase 2 stage. In this, the sister chromatids. So these two are which are represented here are the sister chromatids. Earlier, the separation of the homologous chromosome was happening. Now the separation of sister chromatids is happening in the metaphase, like in the meiosis 2. So the cohesin which is present in between the sister chromatids get proteolytically moves to the opposite poles. So these are the stages of the meiosis 2, in which starts with the prophase 2, followed by metaphase 2, anaphase 2, and cytokine. So in what happens in prophase 2 is a new chromosome. So in meiosis 2, these are the 4 different stages of meiosis 2, prophase 2, then metaphase 2, anaphase 2, and telophase 2, and cytokinesis. So in prophase 2, a new spindle forms around the chromosome, as we can see. And in the metaphase 2, these two, so this is the results of the meiosis 1. So after this, this is happening in both the cells. So a new spindle forms around the chromosomes, and in the metaphase 2, these chromosomes get aligned at the equator, and in the anaphase 2, these two sister chromatids get separated from each other, moving towards the opposite poles, and by the telophase 2 and cytokinesis. So in telophase 2 and cytokinesis, finally a nuclear envelope forms around each set of chromosomes, and the cytoplasm divides, resulting in 4 haploid cells. Right? So this is happening in the meiosis 2. So if we have to observe the complete stage of meiosis, it starts with the interphase, where the cell prepares itself for the meiosis. Then in the prophase 1, there is a formation of chiasmata, and there is a genetic recombination that happens. In the metaphase 1, there is an alignment of homologous chromosomes. So it helps. So there is an alignment of homologous chromosomes. And in anaphase 1, there is a separation of homologous chromosomes. Then in the telophase 1, these 2 homologous chromosomes get divided into the 2 different cells. So this telophase 1 is further followed by prophase 2, where now there is a separation of sister chromatids. So in prophase 2, the sister chromatids, the cell prepared itself to divide the sister chromatids, and in the metaphase 2, these 2 sister chromatids align. So there is an alignment of sister chromatids. In the anaphase 2, there is a separation of sister chromatids, and in the telophase 2, the sister chromatids get completely separated, and there is a formation of furrow, and ultimately, which is followed by the cytokinesis, resulting in the 4 haploid cells. So the 4 haploid cells are formed. So from this 2N cell, there is a formation of 4 haploid cells. So this happens in the meiosis. So this is the complete meiosis. Now, what is the significance? As I mentioned, it is the mechanism by which the conservation of across the generations. Because if the reduction of chromosomes does not happen at the earlier stage, then the in sexually reproducing organism, there will be the duplication of the chromosome in every further generation. Therefore, this process helps in the reduction of the chromosome number by the half, and therefore helping in maintaining the number of specific number of chromosomes across the species. It also increases the genetic variability by the process of recombination, as we see that happens in the prophase 1. So variations are important, very important, and for the process of evolution. So variations are important for the process of evolution, and we know that this variation may happen either at the prophase 1 stage or even during the metaphase 1 stage when there is an alignment. So there is a tendency to form numerous So there is a tendency to form the numerous possibilities for the arrangement of the DNA, and depending upon the arrangement of the chromosome, the that chromosome will be transferred to the next. There is a numerous possibility for the formation of gametes, for gamete formation, right? So therefore, through these mechanisms, variations are being induced. But one of the very important reasons of the variation is the recombination, where there is a recombination that happens between the non-sister chromatids of the same homologous chromosome. Now, so prior to both mitosis and meiosis, diploid G2 cells contain pair of homologous chromosomes, with each chromosome consisting of 2 chromatids. During mitosis, the chromatids of each chromosome split apart and separate into 2 daughter nuclei in a single division. As a result, a cell which is produced by the mitosis contains 2 pairs of homologous chromosome and are genetically identical to their parents. So it remains the same in mitosis, but in meiosis, the 4 chromatids of a pair of replicated homologous chromosomes are distributed among the 4 daughter nuclei, resulting in the 4 haploid cells. Meiosis accomplished this by incorporating 2 sequential divisions without an intervening round of DNA replication. So this is important. DNA replication does not happen again. And 2 sequential divisions. So meiosis happens by incorporating the 2 sequential divisions. In the first meiotic division, each chromosome consisting of 2 chromatids is separated from its homologue. So there is a separation of the homologous chromosomes in the first division. So as a result, the daughter cell which are formed in the first meiotic division contains only one member of each pair of homologous chromosomes. So they will contain only one member or one pair of each pair of homologous chromosomes, right? So there is a separation of only the homologous chromosomes. For this to occur, homologous chromosomes are paired during the prophase of the first meiotic division, and it has no counterpart in mitosis. Now, when they are paired, homologous chromosomes engage in a process of genetic recombination, as I mentioned, that produces the chromosomes with new combinations of maternal and paternal alleles. So alleles or the genetic material get exchanged and resulting in the new combinations. In the second meiotic division, there is a separation of the sister chromatids. So the 2 chromatids of each chromosomes are separated from one another. So there in the second meiotic division, there is a separation of sister chromatids. So this is about the meiosis. In meiosis, what we see is it is a reductional division involving meiosis 1 and meiosis 2. The thing which we need to remember is that meiosis 1 involves the separation of homologous chromosomes, which has no counterpart in the mitosis. So there is no separation of homologous chromosomes in mitosis, but in meiosis, and which is followed by meiosis 2, which is similar to mitosis, where there is a separation of sister chromatids. And the separation of these sister chromatids now will result in the production of 4 haploid cells. And as compared to the in mitosis, we have the daughter cells, which are genetically identical to the parental cells and are diploid. So separation, so these are some practice questions, like separation of sister chromatids in meiosis occurs during the which phase? So here we are separating the sister chromatids. So we know that sister chromatids get separated during the meiosis 2. And which phase of meiosis 2? The separation happens in the anaphase. So answer will be anaphase 2. Second question is, which of the following division results in the reduction of number of chromosomes? So we know that which of the following division results in the number of chromosomes. So it is the meiosis 1 that leads to the reduction in the number of chromosomes. In the meiosis 1, there is a separation of homologous chromosomes, resulting in the reduction of the chromosomes. Next is cytokinesis in meiosis 2 is followed by the formation of, so it results in the formation of tetrad cells, that is the 4 haploid cells are formed, right? The next is meiosis can take place in prokaryotic cells, haploid cells, both haploid and diploid, or in diploid cells. So we know that meiosis is a reductional division and it takes place in the diploid cell, where it leads to the conversion to the haploid cells. The next question is which of the following does not occur in anaphase 2 1 but occurs in the anaphase 2? So it is the condensation of chromosomes, poleward movement of chromosomes, shortening of spindle fibers, or the splitting of the centromeres. We know that the splitting of centromere resulting in separation of sister chromatids happen in the anaphase 2 but not in the anaphase 1. So the next question is, if a cell had 10 chromosomes during prophase 1, how many chromosomes will be present in the late anaphase 1? So in the prophase 1, if it has 10 chromosomes, now the separation will happen and it will have how many chromosomes? Now it will have the 20 chromosomes. Fine. So this is the next question is, calculate the value of A and B. So we know that in the meiosis 1, the reduction happens. So if this is meiosis 1, this is meiosis 2. So in this, the A will be 24 and B, since it leads to the formation of haploid cells which are identical by the separation of sister chromatids, the value of B will be 2. So A will be 24, which upon reduction gives the value of 12, and this upon further the separation of sister chromatids will lead to the value of B, which is 12. How many haploid cells are formed at the end of meiosis 1? So how many haploid cells are formed at the end of meiosis 1? Only 2 cells are formed, and if it is asked about the meiosis 2, it will be the 4 haploid cells. The last question is, which of the following is correctly matched? In the leptodyne, there is a formation of bivalents. In the diplodyne, there is a chiasmata that appears. In the pachytene, chiasmata terminalization, or in zygotine, which is a formation of bouquet. So the correct answer is diplodyne, where the chiasmata appears, and this chiasmata is the result of the formation of the crossover of the non-sister chromatids of the homologous chromosomes. So with this, I would like to end this chapter. Thank you all for attending it.